Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course on computer-aided drug design. We will continue on the topic of target-based drug design. That means looking at the docking of small molecules to the target. So the target could be an enzyme. So the drug could be an inhibitor. It could be a reversible inhibitor, irreversible inhibitor. Okay, target could be a receptor. So the drug could be an agonist or an antagonist. Target could be an ion channel, so you could be designing drugs as ion channel blocker. The target could be a transporter, so we can design an uptake inhibitors. Target could be a DNA, so the drug could be an intercalating agents, minor groove binders, antisense drugs. So depending upon the target, you call your drug as inhibitor or agonist or blocker or uptake inhibitor or intercalating agent or minor groove binders and so on actually. So this is how. So if you look at the enzyme, you could have a competitive inhibition, non-competitive inhibition, allosteric and so on. So look at this. Your enzyme, you must have all studied in your biochemistry or enzyme technology course. We have an enzyme, we have the active site, and then we have the substrate. Okay, so the enzyme substrate complex is formed. Okay, the enzyme substrate complex is formed, as you can see. Okay, then it's producing the product. Okay, this is how things happen here. And so when you have a competitive inhibitor, the inhibitor goes and binds to the active site here. Okay, so it forms an enzyme inhibitor complex. So the substrate is not able to come and bind. Okay, this is taken from this type of place. Reference. And so this is a competitive because your drug is competing for your active site like your substrate. If you look at allosteric inhibition or non-competitive inhibition, what is happening? The inhibitor may be binding to some other site. Okay, not the substrate site, some other site. It's called an allosteric site. By doing that, the active site gets disturbed, or the structure of the enzyme or protein gets disturbed, so the substrate is not able to bind to that. So that is another approach by which drugs can act. So drugs can go and bind. For example, sulfur drugs binds to the active site of where the substrate like para-aminobenzoic acid goes and binds. So para-aminobenzoic acid is not able to go and bind, so sulfur drugs are competitive. Whereas if you take some other drugs, some other antibiotics, they go and bind as an allosteric inhibitor, and hence prevent the activity of the enzyme and also prevents the binding of substrate to the active site. Okay, so in our target based approach, what do? We identify the target. Okay, that means we know the disease mechanism, we know which enzyme I want to target, or a receptor or ion channel. Then we get the determine DNA and protein sequence. Okay, two-dimensional sequence like we looked at. Then look at the elucidate structure and function of the protein. Prove the therapeutic concepts in animals, develop assay for high throughput screening, mass screening, and then select one or more lead structures. Okay, so if I know the target protein, I know the active site, and so I design or I screen large number of molecules so that they go and bind very efficiently. So it is like finding a key for the lock. So we test hundreds of keys and find that one particular key nicely fits into it and opens the lock. So life is very simple. I have the three-dimensional structure of my protein or target. I know the active site also, but life is not always so simple. The second, so the second approach is I know the protein sequence, so that means I do not know the three-dimensional structure. So what do? I do something called a homology modeling, and then create the three-dimensional structure of the protein of my interest. The homology modeling is created with respect to another protein which has a sequence similarity. We talked about sequence similarity some time back. Okay, it is like building the log from the shape of similar logs. Okay, so there is one log I want to build based on some other similar log. Once I do that, I have here target protein 3D structure is built. Okay, then I make the key. That means again I perform the ligand based docking and identify the key. Okay, so that is, so I build the 3D structure of the protein target based on the protein sequence and based on similar protein structures, and then I build the key. The third approach could be I have a lot of ligands which seem to work on the particular protein. It is like I have a lot of keys which seem to open the lock. Okay, so I get the 3D structure of the active site of my binding based on the structures of all these keys. So I have a lot of keys, and so based on the keys shape and size, I will try to predict what will be the active site shape and size. So that could be another approach. So these are all different approaches one follows in the target based design. So this approach is generally common. We may have the two-dimensional sequence, amino acid sequence of the protein of our interest. So we prepare the homology 3D structure of that based on sequence similarity. Once I have the 3D structure, I do a refinement, and then I go into the docking of various ligands from database into the active site of the created protein. Okay, so there are many softwares, commercial softwares, non-commercial softwares are there. Okay, Autodoc is one of the best. It's free for academia, one of the best actually. Then we have the doc, which is also free for academia. Then we have Biosol, FlexX, no. Fred S, Glide, no. Gold again, no. ICM, no. So there are many commercial softwares which we need to pay. They may have different commercial rates for academia and industry, but there are free softwares like Autodoc is one of the best softwares which can do the docking, and it's widely used. So we can use this software, and I will also show you demo of Autodoc as we go along. Okay, so Autodoc, this is the HTTP Autodoc scripts edu. This is a suite of automated docking tools. I will show you later. It is designed to predict how small molecules bind to a receptor. It can calculate binding energies. Okay, so we have Autodoc 4, Autodoc Vina, which can do the job. Then we also have SwissDoc, www.swissdoc.ch. This is another free software which can do the job of docking, and okay, both are very good softwares which we can use. Okay, and I will, as I said, I will show you some examples of how to do docking using both Autodoc and SwissDoc. Okay, this is Autodoc. Okay, so we can download the Autodoc version 4, and for academia, as I said, it's free, so we can use that without any problem, and it's one of the best. We also have something called SwissDoc, and that's again, it's a free software. It can do the docking. Okay, as you can see, we can do the docking. We can give both the ligand. Here we can give the target protein, here we can give the ligand, and then we can give your job name, we can give the email ID, and then we get the results. Okay, whereas in Autodoc, once you download, it's a standalone, whereas SwissDoc is through a web server. Autodoc is a standalone, we can download into our computer, then set it up and then run it. We can get files, docking log files, DLG files, which gives you the results of your docking. It gives you all the non-bonded interactions, it gives you the binding energy and so on actually. So it's a very powerful tool. Now if you look at homology modeling, homology, like I said, is the most important thing. We need to look at the two-dimensional sequences of the protein of your interest with other proteins found in the protein data bank. What is the concept of homology modeling? So this information is available in this PDF. I'm just giving you a summary of that. During evolution, the structure is more stable and changes much slower than the associated sequences. Okay, so the structure remains almost the same even if there are small changes in the sequences. That's the finding from evolution. So similar sequences adopt practically identical structures, and distantly related sequences still fold into similar structures. So that means if the similarity is even 30%, 40%, we can say the structure, three-dimensional structure will be almost the same. Okay, so even if sequences change, the structure is more stable. This was first identified by Chotia and Lesk, later quantified by Sander and Schneider. Okay, so if the length of two sequences and the percentage of identical residues fall in the region marked as safe, the two sequences are practically guaranteed to adopt a similar structure. Okay, so if the number of aligned molecules is 150, then this region we can call safe homology model. Okay, so percentage of identical residues, number of aligned residues. So if I have 100 and I get about 30%, so chances are the three-dimensional structure will be almost right. So two sequences are practically guaranteed to fold into the same structure if their length and percentage sequence identity fall into the region, in this region. So for example, if you have 150 amino acids and if it is 50% percentage sequence, that's fantastic. You can say they both will have a similar structure. Even you can go down to 35%, still we can call it they will come to the same structure. So in homology modeling, what is it done? So we want to know the structure of sequence A, 150 amino acids long. So we compare the sequence A to all the sequences of known structures stored in the PDB using the BLAST command. We just looked at the BLAST command. Now we find a sequence B which has 300 amino acids long, containing a region 150 match exactly. Okay, 150 match exactly. So the sequence identity is 50. Sequence A is called the target, sequence B is called the template. So we build the structure of A based on the template B. Okay, that is what this. Once we do the BLAST, in the previous class I showed you how to run the BLAST. Okay, so if you want again, I'll show you the BLAST here using okay, so when you run the BLAST, for example, I ran for a 5-lipoxygenase BLAST. Okay, it tells you what are the various sequences, protein sequences which match, starting 100%, 98%. You can see different from 5-lipoxygenase from different animals, gorilla, macaque, and so on. Okay, so you can see quite high. So we can take any one of these proteins, which we call it the template, and then build the target protein. Okay, and as mentioned before, the concept of evolution, that if the sequence identity, percentage identity is greater than 35-40%, chances are they will adopt similar structure. So we can get the target. So even any one of these proteins also we can take, because they are very high sequence identity, as you can see. So the BLAST is the first step which we run to identify what are the various proteins which have sequence identity, very high sequence identity. As you can see here, it goes very high. Okay, so that is called the template and the sequence. Using the sequence A, you are building using the template, so that is called the target. So we do the template recognition, initial alignment, alignment correction. So we have used the backbone. Then we do the backbone generation, loop modeling, side chain modeling, model optimization, model validation. All these things are done when you do the homology modeling. So first you compare the sequences, then we do the homology modeling. Okay, so for example, we can Swiss model software. Okay, it's a very good software for homology modeling. It does a good job. So we can look at Swiss model. Yeah, Swiss model. You can say start modeling. Okay, so we can give the upload target sequence. Okay, we can upload target sequence, then search for templates. So we can upload the target sequence. This is a prostaglandin H2, PGH2, GH2 synthase. Okay, which for the mouse. So we can input that. Okay, so this is the sequence of the target. Then we can search for template. Okay, so it's doing the job. Okay, template results. So it keeps doing that. Take a little bit time. Okay, so once it does, it gives you the result here. Okay, so it says sequence identity. We have a prostaglandin synthase, another template, another template like that. It gives you the sequence identity also. It also tells you how the structure was obtained using X-ray with the resolution of 2.1 angstrom, 2.83, like that. Okay, it takes a little bit more time, so it's taking time. So it gives you a lot of things. So we can select one of them as our template, and then model your system. Okay, so if you click on any one of them, you get the structure, 3D structure of your template. Okay, it takes time here, as you can see. Okay. Okay, we can do for any protein. Again, look at this protein. This has got a UniProt number of. Again, we can run this also, no problem. And then this protein, start modeling, so we can give the UniProt number. Now this protein is 5-lipoxygenase, human 5-lipoxygenase, which converts arachidonic acid into 5-HETE. Okay, so we can look at other proteins, so we can give that also. Okay, so this is the sequence, and then we can say search for templates. Okay, this is the target, this is the template. In the previous also, okay, it's still running for prostaglandin E synthase. Then you can say search for template. Okay, so it can do that also. So this also will take time to come. Yeah, it takes time. So as you can see, it's doing it, predicting alignment, state conservation. So it keeps doing that. It takes little time because it's going to look at large number of proteins, and then see how the sequence comparison is, and then we can select one of them. So we will do that. We'll continue. Okay, so if we do for the 5-lipoxygenase, you may get a series of proteins, okay, with different percentage identity, as you can see here. Okay, the three-dimensional structure, how it is determined, X-ray, crystallography. With this is the method used. Okay, what are the ligands present? It contains iron, because lipoxygenase contains iron also, and then I think it contains arachidonic acid, arachidonate here, and so on. If you look at the previous one also, the prostaglandin synthase enzyme, PGH2, it also gives you different ligands that are found here. Okay, it's a dimer, and if you look at here, this is mostly predominantly a monomer. So it gives you a lot of information. We can select the protein of our interest. Okay, so yes, we got some results here. When we try to align the sequence of prostaglandin GH2 synthase, of course its own one, we can look at this one also, for example. So we got this protein, as you can see here. This is the protein. As the template which is selected, so we can do a homology model of with that protein if you want. Then we can say build models with this particular protein. Okay, build model with this protein. So that will be our modeling job. So that is your template. Your target is your original sequence which you gave. So it will build a homology model, as you can see here. Okay, so here also it's given for the 5-lipoxygenase. Okay, so it's given for the 5-lipoxygenase. As you can see, there will be an iron, because the lipoxygenase contains iron. Okay, so as you can see here, the iron is here, iron is here, there. Okay, okay, so in this case we have said build model, so it will start building models. We will see that later, a little bit later. So the first step is aligning your sequence with the known proteins in the database. Okay, so we can use this particular software. Is very good. Swiss model software. Once we take the best one which we feel, and then we can say build a model. So we get a homology model with that, using that template. Okay, so for example, so homology model, as you can see here with that template, which has got an arachidonic acid also inside this. Okay, so okay, so it's built a model. So it's built a model. It tells you how the quality of the model. Okay, it tells you the quality of the model, both the local similarity with the target and the template, and then it compares. Okay, number of residues has been compared. It gives you Z score. Okay, so it compares your model, target model, with using the template, and so we got a model. Okay, based on the two-dimensional sequence. So Swiss model is a very good software. Okay, it makes use of to identify and get a TD structure. There is also another software that's called target prediction, and that's called using a Swiss target. Given a ligand, it tells you what are the possible targets for the ligand. Okay, so in Homo sapiens, for example, so here we can place the smiles in this box or draw a molecule. So it tells you what are the different targets possible targets, making use of structure activity type of relationship. Okay, how do I get those smiles? For example, let me take aspirin. Aspirin. So this is the smiles for aspirin. So we go to the target prediction database. So we paste the smiles. Then we say, yeah, this is the aspirin. Okay, as you can see, acetyl salicylic acid. Submit. So it gives you possible targets for aspirin based on the structural similarities. Okay. So this is also a very useful database, because if you are looking at side effect profiles of molecules, we can see what could be the possible side effects, because the molecule which you designed may go and bind to. Okay, so it's given the targets. As you can see, prostaglandin, because aspirin comes in the arachidonic acid pathway of inflammation. COX-1, this is called prostaglandin G synthase 1, synthase 2, COX-1, COX-2, it binds to both COX-1 and COX-2 enzymes. And then other sodium dependent, okay, transporters. So it gives you many other possible, mostly transporters. It goes to a lot of transporters here. Enzyme, predominantly 33%, okay, and then transporters 47%. So it can go to a lot of transporters, as you can see. So this is also the server is also very useful, because it gives you possible targets. So probability almost greater than 50% here. You can see here. Of course, the COX-1, COX-2 100%. It's well known. Other transporters 50%. So there could be a lot of transporters into which this your aspirin may go and bind to. So this software is also very, very useful for us to make use of as we go along. Okay, so we have seen many softwares like homology models, and then building your target 3D structure, making use of the template. So we need to know how do you make use of the docking software, especially the AutoDock. So we will look at how to make use of AutoDock in the next class, because AutoDock is one of the best softwares which can look at ligand protein docking. Okay, once we have the three-dimensional structure, so we can make use the homology models softwares, okay, homology model softwares, or we can use the Swiss model softwares to align the sequence of your protein with the known protein 3D structures, and then with those templates we can build the 3D structure of your target, and then get a good structure, and then go to AutoDock, and then see how the docking takes place, and we can also estimate the binding energies and so on actually. So we will continue more on this topic of docking in the next class. Thank you very much for your time.